cycle in a cell. So in our body, if you talk about individuals, we have many cells, and the cell can be cycle, and then it duplicates. The way they do is from a parental cell, and they're completing a different phase of cell cycle, then give rise to two offspring cells. So one might be, these are symmetrically divisions, but some of the cells, for example, the stem cell, actually are using the asymmetry divisions in a way, but they all divide from 1 to 2. Yeah, that is how our cell divide and propagate it. So a parental cell will first enter from a G0 phase, go into a G1 phase. So the G is stand for the gap in between S phase, which is a DNA synthesis phase, which stands for this S right here, DNA synthesis. So your genome will be doubled in this S phase, then went through the G2 phase, go into the M phase. M is so-called mitotic phase. Mitotic phase is when you have the duplicate chromosome, then you separate the chromosome into two daughter cells. It's a mitotic phase, what they do, and end up after separation, they will have cytokinesis. Then the cell will divide into two cells. So the gap phase is try to prepare the material that are ready for the next phase. For example, G1 phase will prepare a lot of protein machine required for the synthesis of the DNA, which is, for example, the DNA polymerase. And also there's other material, for example, your adenine, your G, and your C, your T need to be synthesized in different pathway in order to prepare for the duplication of the DNA. And for the M phase, you need a G2 phase to prepare. For example, you need a machine to segregate the chromosomes. And those machines, also your histone need to be duplicated as well. So the histone will be also abundantly prepared in the gap phase as well. So in different phase, there are different checkpoints to make sure that you are properly prepared to enter the next phase. So that is so-called checkpoints. Just like when you go into some of the restriction area, for example, you go to the presidential office, there will be a policeman who will stop you and check your permits, whether you actually apply permits for that or not. And that is a checkpoint. And in the cell cycle, there are cyclins. For example, in this case, in the S phase, there are cyclin A, and they are supporting of like P21, which is a cell cycle inhibitor. And you have a cyclin-dependent or cell cycle-dependent kinase, so-called CDK. Different CDK compositions will make up different checkpoints. So you have different type of policemen checking on different gates, and also when we entry different phase of the cell cycle. So why we need to do that? Two ways. One is check whether you are ready to go or not, to go into the next phase. The other thing is to check whether you actually your cells or the genome is damaged or not. If it's damaged, you need to prepare, ready to go in a way. So if you cannot pass the check point, then or you cannot repair sufficiently, then cell will choose to go out of the cell phase. Then when you go out of cell phase, then you will either arrest in a stage, so-called, for example, senescence or other phase, or the cell will die and become cell death. So they will become different cell phase, and they will go to a different fate of the cells. So it's very important to balance this. Once you synthesize or you are synthesize things, and make sure you synthesize the right things to maintain the genome integrity, so that then you don't need to repair. if you have made some mistake, or there are certain outside pressure come in, then you need to repair it or restore your genome into the right compositions in order to pass on the right informations. So those are the repair and also the response to the when your genome is consult by others different stresses. So I will give you some idea about what type of damage from outside, also inside from inside. They are so-called endogenous or exogenic, or intrinsic or extrinsic pathways that are leading to DNA damage, and then also leading to different repair pathway or different cell response. If you didn't properly respond to this type of insults, then you will suffer from the genome damage. And the genome damage is leading to misreading of the genome information, now causing a dysfunction of your cell and leading to dysfunction in your organs and leading to the disease occur. Okay, and today I will give you some example of the disease, mainly focusing on the aging process and also the disease of the cancer. Okay. So the DNA repair in 2015, we actually just announced this Nobel Prize winner this year. And the most case actually should congratulate to the Kyoto University of the PD-L1 in an immuno checkpoint and also immunotherapy for the Nobel laureate winner, Hongzhou Sensei. So this is in 2015. It's the year of the repair. In 2015, there's 3 winners of Nobel Prize in chemistry on repair, their contribution to the repair. For example, the Thomas Lindau is famous with his repair on like glycosylase, the excision repair pathway. And Paul Modrich is famous with his discovery on the mismatch. Say, for example, if you notice that the genome, for example, maybe I can ask the Kyoto sites. If you know, if you have your DNA is composed with different purine and pyrimidines, you have A, T, C, and G. And usually A will match with what? Any side? Anybody from Kyoto side?
T.
T. And C will match with
G.
G. Okay, so if you pair A to G, what happened? What do you think?
If you might end up with a mutation later on.
Okay, you're leading to mutation. That means your DNA structure is unstable. Okay, those are genome instability. And A and G are mispair. That is so-called mispair or mismatched. And then you need to repair this in order to avoid the mutation or the genome instability to occur. Okay, so that is the mismatch repair in brief, so-called MMR. Okay, then the excision repair can also do the things as well, but they can repair different type of mutation. And those are, if you are at the nucleotide level, it's so-called nucleotide excision repair, and which is in brief as a NER. If you repair at the base level, then will become base excision repair. That's so-called BER. Okay, so there are shorter patch and also longer patch of different type of repair pathway. And the final one is the Aziz Sinka, which is also from United States. And he's the one who actually discovered on the nucleotide excision repair machine. Okay, and below is 2 professors in the College of Medicine here at Taiwan. One is Fang Weihong, who's in the medical technology department of the College of Medicine. and J.J. Lin is at the biochemistry and molecular biology department of the College of Medicines. And Professor Fang is Professor Paul Modrich's student. And if you read in the textbook and you read into the nucleotide excision repair, you will see the Azizinca and you'll find out the UBRAB in the E. coli. That's the work of Professor Lin when he was a PhD student at the Azizinca's laboratory. So he worked out all the biochemistry of how the nucleotide excision repair workouts. Okay, so these two professors is currently in Taiwan, and I hope they maybe someday they'll also win a Nobel Prize as Kyoto University professor does. Okay. So, as I mentioned to you, there are different ways of the DNA might be stressed or get the damage from. They are from the outside, so-called exogenic, or so-called extrinsic. Okay, so you can receive, for example, the sunshine. Sunshine has, for example, UV, and UV have energy, and you have a shorter wave, and the energy will be higher, and that will causing DNA damage. And in a form, predominant form will be, for example, the thymidine dimer, which we call TT dimer. Okay, so you have UV light exposure. Sorry. Then you will have chemical damage. Okay, and chemical damage, for example, when you eat things, there are different so-called mutagens or carcinogens, which is chemically more reactive. Okay, or when you go shine with the UV and you go to take, for example, take an airplane, and you are more exposed to the radiations from the universe. So ionization radiation is also can be outside. These are physical damage that can be causing. Endogenously, when you synthesize DNA, chances are, for example, like 10 to the minus 9, you have a replication error. And that will causing, for example, mismatch. So a lot of mismatch is coupled with the replication error when they are doing that, okay? And you can have mitotic, when you have segregations failure, then you will have missegregation. That will be a chromosome level or lost. Okay, so there are different level of the genome information can be suffered to the instability, and we can divide this type of things according to the mutation type. For example, if you are in the nucleotide level, then you have so-called microsatellite instability, we'll call like a MINT. And if you have chromosome level, then you have chromosome type of instability, which we call CIN, C-I-N. Okay, so metabolites inside the cells were also creating different type of reactive metabolites that were also challenge to your DNA as well. So we will suffer from different type of damage sources, okay, and you're leading to different type of DNA damage, and the cell will respond. And if you didn't maintain well, then the genome will suffer to instability and causing to disease or even the cell death or the human individual death, okay? And these are different pathways. So what dictates, what determines, or what decides which repair or which response that need to be activated in order to do that, to correspondingly activate it. So the thing is, in the chromatin in humans or eukaryotic cells, we have chromatin. So we have protein, we have DNA, so all your genome is composed with chromatin. And the chromatin structure alteration is the source. And different type of damage were leading to different alterations or different changes in DNA structures. Let me ask Tsukuba sites. Do you know our DNA, do you know whether it's a helix? And what type of helix property you can describe to me? Anybody in Tsukuba side? Hi.
No, because Tsukuba is not attending here. So Tsukuba side.
Okay. So Kyoto side.
Oh, Kyoto. Okay. Somebody.
Yes. Anybody?
It's double helix.
Double helix. Okay.
Backbone.
And do you know the handedness? It's right-handed or left-handed?
Not sure. Right-handed.
Yes, you are right-handed, right? Okay. And so, do you know the base composition? Your classmate actually tell you that A match with T and C match with G, right? And those base will have creating a so-called stacking forces. And do you know how many base per turn they will turn into right points? For example, from here to here, it's one turn, which they turn right-hand directions for 360 degree. That is defined as a turn. Do you know how many bases in between here to here?
I remember I learned it at college, but I forgot the exact number.
Just a guess.
10?
Or
10?
10? Yes. Okay, that's good. If you cannot count, then you can count. You can count. You can count from 1, 2, 3, 4, 5, right? And 6, 7, 8, 9, 10. So average is about 10. Okay, 10 base pair per turn. Okay, so any structural alteration between, for example, the AT match become an AG match, then we're causing a structural alteration here. And the structural alteration will be different. For example, some of them are not matched well, then they are creating a gap right here. So there's a base alteration by the hydrogen products. Then you have TT dimer, additional linkage. So you tighten the TT together, then you alter the structure. So the structure will be different. And for example, I have my hands, okay? And I have an additional link of TT dimer in my hand. For example, I link this, my two fingers on the so-called index finger and the so-called middle fingers, right? So you have these two are T and T. And the TT dimer, usually they can actually open like this. But the TT dimer have a link. They will link, for example, this is the link of the TT dimer. Then it will become like this way. My hand will become like this. So I cannot open wide as before. For example, before I can open like this, but this. So this is the source of the recognitions. So the repair enzyme will come to recognize this and try to repair back to like this. They will try to remove this and become a hand like this way. Okay? I hope they actually see it. They do? Okay. Okay, so the local structure change determine what the response will be activated. Okay? So you can see there are different forms of different type of mutations or different type of forms of damage, actually. For example, you have the accidents, they will have a G on the A axle G, which is at 8 positions, they will have oxidations. Okay, then you have different type of mismatch created. Okay, so then you have this different sources and leading to different type of damage forms, and they will be recognized. So they will be recognized by the sensor. Okay, so you have the general concept that today you should take back for this lecture. That's the only things that I will ask you to remember. One thing is that you identify the source, where the source is, and how the process will go, and what is the key things that you should know. And the conceptual is that you will have a sensor which to recognize the damage. Then you are leading to the activation of the corresponding right repair. So you have the right repair things to be activated. That will be a mediator, which is so-called transducer. Transduce your signal, your repair signal need to be activated. Then they will process, they will send into different response, and different response will have an executor to do, for example, they will have a repair protein. Those are the effector, or turn off the cell cycle progression, so-called checkpoint proteins. Those are the effectors. So you have three types of player. One is the sensor, which recognize for activation. Typically, it's a kinase. Okay, then you have a transducer, which will be activated by the sensor kinase. So typically they will be phosphorylated. Okay, then they will transduce the signal to the real worker, which causing the cell cycle checkpoints turn on, or the repair machine, and those are so-called effectors. And this process will be amplified throughout the signal transcade or cascades. Okay, so we will give you some example later how those sensors in different organisms, and then the effector will work on different ways. For example, on the repair, or they will work on the transcription to boost the cell ability in repair the whole things. And or if they cannot do anything, they will turn on the cell death pathway, for example, the apoptosis, or they stop the cell cycle transitions. All the things is try to maintain your genome to be a proper, stable stage. Okay, so these are all the things that they want to do. So what you need to remember, you have a sensor, you have transducer, then you have effectors. Okay. So they are leading to different pathways, and what everything is determined by the type of damage. For example, if you have a mismatch, then you have mismatch repair being turned on, or the base excision repair can also do this. And these are from the replication errors. If you have, for example, the ionization radiation, the energy is good enough, and they were cutting the double strand break. Then you have a double strand break repair, which is so-called homologous recombination or non-homologous end-joint repair pathway. So there's a cause corresponding to the damage, then the damage determining the repair pathway. Okay, there are many, many ways which has been studied. Okay. So in eukaryotic cells, it's a naked DNA, or there's a so-called chromatin-like structures inside the prokaryotic cells. But most of people will focus on the DNA type of damage. And most of the discovery actually from the mother organism of the E. coli, or the early days in different type of bacterial cells. So in summary, they usually have three types people will talk about in bacterial sites. There's one is so-called SOS response. So the SOS is a signal which is sending out for helping. Okay, so you have a SOS response, and you have so-called adaptive response, and there's also oxidative response. So these are the common threats for bacterial cells when they are growing on a wild or in a laboratory, that you actually suffer from different type of stress. And different type of stress were leading to, for example, the oxidative stress were leading to the oxidative response to be turned on. They will try to repair the oxidative damage. For example, the AXOG. But AXOG can also leading to, for example, SOS response to activate it. Okay, so there are sometimes they will activate multiple pathways in order to survive, or in order to repair well of the cells. Okay. So I will give you the example of the SOS response to the DNA damage. The key player here for the activation is so-called REC-A. Okay, right here. The REC stands for recombinations. So inside the cells, the recombination is the pathway for try to repair the damage. Okay, in the early days, they define those things, genes are the REC genes. So they have this so-called REC-A gene. And REC-A gene activates the mechanism is through this repressor, so-called LAX-A. LAX-A is a transcription repressor which suppresses the transcription normally. So there are a lot of, for example, repair genes is repressed by LAX-A normally in a normal physiological conditions. Once you have the pathological condition or you have under stress, then the REG-A will be activated, then they will cleave using the protease activity of the REG-A, and then it cleave the LAX-A. So the LAX-A will lose the functions, and then you are de-repress, almost equal to activations of the transcription of the gene that require for repair. So that's the mechanism which is utilized in the SOS response. So that's the key reactions. The REG-A will be activated, then cleave the LAX-A, and then the LAX-A cleavage, the LAX-A will be lost and degraded. Then your gene or the DNA damage inducible genes will be expressed. Then it will control the cell cycle, for example, fix K, or they will also control the sensing pathway, for example, a single strand binding protein or others, okay? So this is the chain reactions of the SOS response. And the key player is the REG-A and also the LAX-A pathway. And they are complement a pathway, for example, the REG-F, REG-M pathway, and the root AB is a resolvase. Okay, so there are many different of the repair pathway can be turned on in here. So these are some example of the recombination repair protein in bacteria. In early days, they all using, for example, the UV to study this, and they all name this type of protein belong to the recombination protein, which is the REG-A, B, C, D. Okay. So there are also a base excision repair being discovered inside the E. coli. And the base excision repair is usually creating a damaged base here. Then you'll try to remove the damaged base here and creating a purinated site. Then the purinated or AP endonuclease will cleave right here and remove a patch from here. Then repatch it or resynthesize using DNA polymerase 1 and ligase to ligate this. So it's sort of look like when you are walking on the road and here has a damaged site. Then you actually try to dig it. You dig here and then you repavement on the surface. So these are the mechanisms of the base excision repair or the nucleotide excision repair is similar. Okay, it's just they are using different machines to do that. So these are the nucleotide excision repair. For example, you have a damaged nucleotide right here, and you have this, then you have enzyme to cut from this side and also this side. Then you have leading to a longer patch of the DNA need to be resynthesized. And they are using the similar DNA polymerase 1 and also DNA ligase here. So then you will religate this. And they are using different enzymes to do so. Okay? And when you have an intra-strand cross-link, they will have a different repair pathway, and they have to remove both sides. And they are using some of the additional enzyme to help out. Okay, but the concept is very simple. You remove the damaged sites, then using the other sites to as the reading template, then you're using the polymerase to precisely and repair this site into the original matching one. Okay. So mismatch repair is very similar, but there's one thing which is tricky in a mismatch. How you know which one is the mismatch? For example, I have an AG mismatch. Do you know original is GC match or it's an AT match? How you decide that? Do you guys know my questions? Yeah. So maybe ask, how about you? What do you think? You don't know? How about you? You can open your microphone so people can hear you. Yeah. Yeah, the microphone is right here. Just say out what you know. It's fine. What I learned is from the book. Yeah. Yeah. Yes. Yeah. Come on. Give me some thought. It's fine. Yeah. Nobody can say you are right or wrong. As long as you speak, you got a chance. You don't speak, you don't got a chance. Yeah. How you determine the AG mismatch, which the G is correct or the A is correct? Yeah. Yes. Yeah. Microphone, using the microphone. Yes. Yes. The hydrogen bond. Okay. So the AG probably has 2 hydrogen bonds. Yeah. So it's more like likely AT is the right one, or GC is the right one. So according to what you thought, yeah. What do you think? Yeah. Your thing is AT or GC? Let's vote. Okay. So if you have a AG mismatch, that's including keratinocytes. If you think it's the GC, it's the right one, raise your hand. Nobody here? Nobody there? If you think it's AT, it's the right one, raise your hand. There's nobody there as well. Nobody here as well. So what you guys think? Or you are still thinking? Yeah. It's very important. You copy the wrong ones. What happened? You make a mistake, right? Sorry. Hi. Hi. How are you?
Me? Okay.
Can you say your name? Yeah.
My name is Crystal. So I'm just basing this off the diagram, but
okay.
The template strand is methylated while the newly synthesized strand is not methylated yet.
Okay.
So based on this, you can identify which base was the original one.
Very good. Very good. So you are very clever and actually looking for the sources. Yeah. That's very nice. These are the things that I admire because a lot of people has different ability. Some people are very sensing to the environment change, and some people can use your surrounding resources very well. Okay, so Aso, or I forget your name. Sorry, is very sensitive in this, and he actually catch the right help from the screen. Okay, so from here, this is the why Paul Modrich get the Nobel Prize, because he solved out the mystery of what I just asked you about it. So if you can figure it out, Paul Modrich maybe just look into certain, like the Newton, you always think about how he figured out the gravity because the apple dropped on his head when he's resting in the apple trees. And many people do that, right? In Chinese, if you are under the apple tree, you probably catch a rabbit. So there are different ways of figure out the theory. You learn from the nature. Okay, so most of the things we learn from the nature. What we are discovering right now, it's already there. There are rules of the nature rules. Okay, so what you are doing is actually just find out or refine out a lot of facts. Okay, so the reason being is that the parental cell usually is methylated already. Okay, newly synthesized. There's no nucleotide which is methylated, then be incorporated. Okay, so the methylation will give rise to the parental cells. Remember, I told you the mismatch usually is follow up the replications. So when you synthesize, you have the template strand, which is the old template, and the synthesized the new strand will be the daughter copy. Then you will have it's usually unmethylated. This is the stage so-called semi-methylated, and so the template strand for synthesis usually is the right ones. Okay, so when you have a mismatch, if here is A, then the G will be repaired. Okay, the G will be on this side. If this is a G, then your A here will be replaced. Okay, so this is actually inside the E. coli. There's a methylation determinant for that. But in eukaryotic cells, nobody really knows. Some people think it's a Nick driven. Some people think other ways. Okay, so these are still a little bit a mystery inside the eukaryotic cells because we don't have the methylation determined there. Okay, so there are different enzymes. I don't want to go to the detail to bother you on this. And so they are discovered, for example, the mute S. The mute stands for mutations. So they are using the mutation or the recombination tool to identify the proteins. And these are the enzymes that are in charge of the mutations. So they call mute. So they are mute S, they are mute L, they are mute H. Okay? So you read into the histories, it's actually quite interesting in this way. Okay? So there's adaptive to alkylating agents, which causing, for example, methylations on the O6 positions or on the T, O4 position of the T. And these were removed by the methyltransferase, and they so-called sort of like commit suicide. They just grab this methylation to themselves, and somehow leading to repair. So these are the methyltransferase function. They are using themselves to grab the damage out by reactions and then restore the G and T from the methylation stage into the normal stage. Okay, so these are the adaptive response into the alkylating agents. These are the examples from the E. coli. So all the principles actually apply to the eukaryotic cells. It's just eukaryotic cell has more complicated genome. Then you have different ways to deal with it. And also we have much mature chromatin structures. So there are chromatin alterations need to be added into these type of pathway. But the basic principle is very similar in here. Okay? And the oxidative stress then will be activated by, because if you read into the periodic table, the O and sulfur is actually in the same line. So they have lone pair of the electrons. Okay, so the reactivity is actually come from there. So the sulfur actually can take over the oxygens. So these are also the natural rule being used in the biology as well. So most of the regulon is using these sulfur or the disulfur bond balance, try to actually remove the oxidative damage causing by reactive oxygen species or reactive nitrogen oxygen species. Okay. So in the eukaryotic cells, there are chromatin structures, and there are chromatin which you know there's H1, H2, H2A, H2B, and H3 and H4. Okay, and it composed of the nucleosome, and these histones can be modified in their tail by phosphorylations. They can be modified by methylation, by acetylations, and causing a different form of change on the chromatin. And acetylation, do you know acetylation? It's positively charged or negatively charged? What do you think? Let's vote. This time you have to give me your answer. If you don't raise your hand, I will ask you a question until the class ends. Okay. Okay. So from the Kyoto sites, if you think the acetylation to the DNA will increase the negative charge or positive charge. If you think it's negative, please raise your hand. There's 3. Okay, you have, I see 6 and I see 3 raise the hand. All girls, I presume. And in Taiwan, if you think it's negative charge, raise your hand. 1, 2, 3, 4, 5. We have 6 people here, and also there are 5 people think it's negative charge. And if you think positive charge, raise your hand. Okay, 1. Okay, so what happened to the other 2? Okay, and how about here? Positive charge. The only one left. I don't know. No? Okay. So it's a loosened form. So it's a more negative charge way. So it's more loosened, and the acetylated status actually loosen the chromatin, so it's more ready to open the chromatin. So it's indication in the transcription that is active transcription regions. Okay. So it's logically, it's how they think about this. So inside, when you want to repair the chromatin structure, you want to lose the chromatin. So make the accessibility higher to the repair protein. Okay, so for example, if you are in here, if you look into here, can you see, I think if they can adjust. So this is the table that you are actually have, right? Okay, so you can see there's a actual microphone here. Can you move to here? Yeah. So this is the microphone, and here. And if you want to work on this track, you are actually hit this so-called microphone, right? And you can change. This way you cannot see it. Okay, so I can actually write through here. Okay, but this is so-called creating the accessibility. For example, I can access here to go through. But if you have something here, this is more tightened. But if you are loosened, they can walk through. Okay. And in here, usually you have, for example, this is a chromatin structure. This is naked DNA. This is chromatin. Okay. So you have nucleosome here, and this creating a blockage of the accessibility. If there's a damage under this bag, I need to remove the bag to create accessibility to loosen the chromatin in order to access to the template to repair. Okay, so this is a concept of the repair, or any machines that you want to work on your template. You have to loosen your template, which is the chromatin concept. Okay, and acetylation can help in doing that. Okay. Okay, so these are some of the concepts with the eukaryotic cell. They are more complicated because they are local chromatin alterations created by the DNA damage. Then it's more complicated because we have more genes. We have tons of other genes, but the concept is very similar. You actually have the signal, DNA damage, and you have a sensor, you have a transducer and effectors. So come again, we have sensor, transducer, effectors, and corresponding to different type of things. And in the E. coli, remember SOS response, there's only RAC-A and LAX-A, which is the key player. But in eukaryotic cells, we have many of the, for example, inside here, this is the sensor kinase, or transducer in between. There's so-called this called PI3 kinase, and there are many of the PI3 kinase. We have total of 5, and 3 of the PI3 kinase is related to DNA damage. One is ATM, the other is ATR, and also the third one is the DNA PK kinase. Okay, and these are all the master kinase. So they will mediate phosphorylation of, for example, the RED9 and RED1 and HAS1. And this is so-called the 911 complex, which is for the rescue, and just the same concept as the SOS response. So then it will activate the transducer and leading to the activation of this effectors like a CHEK1, which is the checkpoint protein. Then it will block the cell cycle progressions. They are leading to, for example, activation of P53, and that will leading to either repair or the cell death. Okay, and this P53 is a transcription factor. They will activate multiple gene expressions. There are also other proteins. Okay, so if you take a glance inside here, so the same thing is that which type of damage you are leading to what type of activation of the DNA damage response. And they are more complicated inside the eukaryotic cells. And you can just take as a reference here. This is 2015 on the Journal Cell Science. And they summarize the ATM and ATR signal at a glance. And you can see there are many factors actually involved inside here. But what is the driven force? As I mentioned to you, in the early phase, when you want to recognize you have sensor, it's the DNA damage determining everything. And what I'm asking is that when inside the ATM, they are actually kinase. So what driven the pathway goes on? It's actually the phosphorylation pathways. Okay, and the phosphorylation pathways, I need to mark the area of the damage. So the recognition is important, and the local modification is important. And when you have a local modifications, for example, here, if you see inside here, these are all the DNA. For example, here, I'm not sure whether the camera can follow me or not. This is ATCG GCTA, for example, like this. And as you can see from here, this line, you will notice this one is modified, right? I have a code here. Say, for example, the code is methylations. Then you will actually modify right here, okay? And then you modify here. Then you'll see this. This is need to be recognized, and you're creating a focus of your attentions. This is so-called focus. And you have a multiple focus, it's so-called foresight. This is where the repair happens. If you label the protein who come here, then you will be able to see where the repair happening. That is so-called repair foresight concept. So you can use in the microscope to see that. You can see right here, you have different points, which is different focus, which is in a plural, it's a different foresights created. Those are the repair pathway. Then you have time A from 1 minute, A come. B will come in 6 minutes, and C will come in the second minutes, the 2 minutes. Okay, then you have a chronic order, and remember what we said: sensor, transducer, and effectors. Which one will come first? What do you think? Sensor, transducer, and effectors. The sequence is that you recognize activated by the sensor, then you have transducer transduce the signal, then to the effector to do the work. The sequence is sensor, transducer, and effectors. And you using couple with this technique of the foresight analysis with the time course, then you will know who is sensor, who is transducer, who is affected. Okay, so these are the experimental logic that I would like to bring to you, and then you can use the technology and you can apply to different technique. Then you can watch them as a play to display inside your eye. It's like into the theater, you go inside, the first thing father come out. If it is a wedding, the father will stand there and receiving the bride come in. So the bride will come later. The bride won't just come in the first play and he stand there, right? Because it has to come in and they has to have a very beautiful and have to make a makeup and everything. So it's like a play. So it has different temporal and timing wise and also space wise, which is spatial position. And you have using the label of the GFP or different cytological marker to watch them come out. They are dressing in different colors and different timing come out. So you can actually draw out a pathway with the cascade and timing, and then you can actually put everything together into the pathway. Okay, so if you are interested, you can go look into the Cell in 2004. They actually using the model organism and also human to play on the whole play, and you can watch them dance in this. Okay, so the driving force is on the phosphorylation, as I mentioned to you. I will just go through for you. There's two type of domain. One is on the tyrosine, the other is on the threonine and serine phosphorylations. And there were different domain being recognized and then passed transduced. And there are different examples for that. For example, focal head activation domains that you have a REC 53 as a threonine kinase as an example. They are different domain and created by different recognition. Okay, then after you turn on, you will turn it off. Then you have this turn off of the DNA checkpoints pathway. Usually, if you have phosphorylations, what happens if you want to turn it off? You take away the phosphorylations. So we have so-called phosphatase. Okay, so you can use in this way. Or I just degrade the activator of proteins. Okay, so you can degrade that proteins and then you turn around and cycle again. So there are different ways of turn off this DNA damage checkpoint. So I think time is almost up. So I want to bring to you just a brief concept of the summary in this. That today you learn damage, what's the determining of the DNA damage response. It's how the DNA damage alter the DNA structures. Okay, that it's the key determining of all the response coming out. Then you have a sensor come in to sense the DNA damage, then transduce the signal to transducer for activation of the subsequent pathway. Then your transducer will deliver the message to the worker, which is so-called effector, causing the biological effect change. Okay, so you have sensor, transducer, and effector pathway, and giving you this. Okay, so these are the things, and once the thing is done, then you will turn off and return to the inner balance. So you need to turn off that. And the key player in here, it's actually some of them are mostly kinase, and the phosphorylation being the key determinant also for the transducing pathway. Okay, so the rest of them I won't go through for you now. These are the more complicated pathway of repair inside the cell. So I want to go to in the end that if you don't repair properly, genome instability or causing, for example, the aging process, like your organ or your wrinkle formation is related to the repair capacity. And when you are getting old, is actually will have the aging process will actually getting accelerated, and because the repair capacity is decreased, and you will also suffer for disease of cancer. And I will continue to talk about this on the cancer development on the Wednesday courses. If you are interested, you can come to medical school on Wednesday 9 to 11 in the medical school. If you are interested to have the follow-up, more detail on how these were leading to the cancer development, also initiations. Okay, so there are different check pathways and being activated parallel pathway, and understanders were leading to the therapy method development. I will cover that inside in the other class of the long distance course. If you are interested, you can come I'm welcome you to come. But you can also read by yourself by the Hannah and Weinberg. They have two versions of the cell cycle, cancer hallmark in 2000 and also 2011. Okay, you can all go into the cell type in the cancer hallmark, then you'll find out. So in the end, I will give you a little bit of the introduction of today's paper, that inside the informations were driven the oxidative stress, and the oxidative stress will activate a DNA damage pathway, and that leading to the genome instability and leading to the genome rearrangement, and it's a common hallmark for the cancer, and that has highly relationship with the cancer formation in the prostate, which I think the male should actually pay much more attention, and it's published in the Cell Report 2016. Okay, so I hope you enjoy the lectures, and I think we should take a break, right? So how long we should take? We take a 5 minutes break, and if you have questions, I will be here. We can just chat. It's fine. Okay. So hope you enjoy. Yeah. Thank you.
Thank you.
Kyoto sites, anything that not clear for you?
Are there any questions from Kyoto side?
Yeah, it's a break, so you can leave at your please. Yeah, but if you have questions, I can answer you right here. Yeah.
Hi, TK. This is James.
Yes.
I just wanted to ask, since you're the expert on, yeah, I'm hiding over Chisaka Sensei.
Okay. Yes.
Since topoisomerases are your specialty,
oh, yeah, okay.
Can you tell us just in two seconds, how do topoisomerases affect genome stability?
Okay.
What happens if they're defective?
Actually, the paper is why I choose this paper is highly related to the topo property. Inside the topo, in the prostate cancer, you know, the androgen is the key determinant for the maleness of your body. For the female, for the male, you have higher androgens type of hormones, which determine your male figures and so on. And those are governed by the AR, androgen receptor mediated transcription activations. And this transcription activation is highly related and regulated by topo2 beta isosign. But if you have this oxidative environments, this oxidative activations will trigger the topo to become a nuclease. It just cut the DNA and no religations. That's our current hypothesis. We are studying on this end, and that leading to the genome rearrangement in the chromosome level, and that is causing by the chromosome instability. And that if it happen on the oncogenes, then you activate the oncogenes. It happens on the suppressor genes, tumor suppressor gene, then we will turn off the tumor suppressing functions, and now leading to the tumor formations. And that is one example I can give you on the topoisomerase once they go bad, because they cut the DNA and they also rejoin the DNA. And if you turn off the rejoin function by oxidation on the enzymes, and that will leading to the problem of the genome instability. Did I answer your questions, Jim?
Yeah, very good.
Okay, thank you.
I knew I would get you started.
Okay. So so you guys are okay to start? Okay. So the students, you should turn on. Okay.
Okay, so hello everyone. Today I'm going to talk about this paper about inflammation-induced oxidative stress mediates gene fusion formation in prostate cancer. This is published in 2016 in Cell Reports, and my advisor is Professor Lee. And my name is Zhang Haofen. So before we start, let's take a look at the important genes. This here. First is TMPRSS2 is an androgen regulated gene encoded transmembrane protease. And the second gene is ERG. It's an oncogenic erythroblast transformation specific transcription factor. And also the important cell called LNCAP is an androgen sensitive human prostate endocarcinoma cells derived from metastatic site. So let's get started. This study mainly talks about the link between inflammation and gene fusion in prostate cancer. The authors have a hypothesis that inflammation-induced oxidative stress, also called ROS, plays a key role in TNF-alpha-induced TMPRSS2 and ERG gene fusion formation, and androgen signaling is required for this process. They have done several tests, including both in vitro and in vivo, to prove this hypothesis. So let's take a look. First, they stimulated the TMPRSS2 ERG gene fusion negative LNCAP prostate cancer cells with TNF-alpha for 48 hours. They observed robust formation of the TMPRSS2 and ERG gene fusion transcript by TNF-alpha stimulation using TechMan-based qRT-PCR and gel-based RT-PCR. They also did a sequence analysis of the TNF-alpha-induced fusion transcripts. It revealed joining of the first exon of TMPRSS2 with exon 4 of ERG, which is the most common TMPRSS2 and ERG isoform in prostate cancers. Next, they examined the role of androgen signaling in this process. They found that depletion of androgen from the media or androgen receptor knockdown by siRNA blocks TMPRSS2 and ERG gene fusion formation upon TNF-alpha stimulation. In addition to LNCAP cells, LAPC4, an androgen-sensitive cell, has the same result. However, the androgen-insensitive D145 cells are resistant to TNF-alpha-induced gene fusion formation. So they made a small conclusion. The genomic rearrangements are non-random, and the cell type-specific events detected by lineage-restricted cell signaling pathways. And the second, intrachromosomal rearrangements are more prevalent than interchromosomal rearrangements. Then they use split signal-based dual-color fluorescence in situ hybridization, also called FISH assay, to observe breaks in the TMPRSS2 and ERG locking upon stimulation of LNCAP cells with TNF-alpha. They found that TMPRSS2 is more prone to DNA breaks in comparison to ERG. And they also study LNCAP cells treated with PBS or TNF-alpha. and they observed robust formation of de novo genomic rearrangement about TNF-alpha stimulation. However, with a defective homologous rearrangement in the absence of an external stress, is relatively inefficient in the formation of genomic rearrangements. A combination of TNF-alpha stimulation and homologous rearrangement deficiency further increased the formation of de novo genomic rearrangement. Then, because TNF-alpha stimulation has been reported to increase reactive oxygen species, the RS production, they hypothesized that oxidative stress-mediated DNA damage may be the underlying mechanism for the generation of DNA double-strand breaks required for gene fusion formation. In this test, you can see the red plot is the control and the blue plot is the treatment. They observed that stimulation of LN-cap cells with TNF-alpha did result in a dose-dependent increase in RS production, like here. And it is enhanced by cyclohexamide, which blocks NF-kappa B-mediated antioxidant production. But it can be reversed by NAC, which is the RS scavenger. They also observed TNF-alpha induced RS production and DNA damage in the androgen-sensitive LAPC4 cell, androgen-insensitive D145 cells, and being internalized non-transformed RWP1 cells. They conclude that androgen signals is not required for TNF-alpha mediated formation of DNA breaks. And rather, they found that androgen receptor redirects RS-mediated DNA damage to the TMPRSS2 locus by enhancing transcription, opening up the DNA, and thereby making it susceptible to DNA double-strand breaks. Second, they found that TNF-alpha-induced DNA breaks in the ERG locus are likely to be androgen receptor independent. They also do another test. They found that although single-agent treatment of LN-cap cells with either TNF-alpha or CHX, which is the cyclohexamide, induced DNA damage, a combination of TNF-alpha and CHX markedly enhanced the extent of the DNA damage, as visualized by phosphorylated histone H2A.X elevated, and also increased TMPRSS2 ERG gene fusion formation. Combined with the result that treatment with NAC blocked TNF-alpha induced TMPRSS2 and ERG formation, they conclude that ROS is the mediator of TNF-alpha induced TMPRSS2 and ERG gene fusion formation. And to confirm their hypothesis, they treat with the H2O2 resulted in a dose-dependent increase in DNA breaks, and TMPRSS2 and ERG gene fusion formation also confirm this conclusion. Thus, they explore whether gene fusions can be induced by inflammation in an in vivo setting. They use a well-studied murine model of in vivo inflammation, as the picture shows here. First, they raise a sterile air pouch in the mouse dorsal, and they will inject stimulus. Is the inflammatory lipopolysaccharide, and the control is injected by PBS. Later, they will inject LN-cap cells, and then they will collect the cells and analyze them. They observed the result in control or LPS-treated air pouches raised in SCID or B6 mice. They did not observe TMPRSS2 and ERG gene fusion transcript in LN-cap cells recovered from the air pouches of SCID mice with or without LPS treatment. But TMPRSS2 and ERG gene fusions were observed in LN-cap cells injected into the air pouch of B6 mice, the levels of which were further accentuated upon treatment with LPS. This result suggests that Analogous to their in vitro data, inflammation in vivo induced the formation of TMPRSS2 and ERG gene fusions. They also did a split signal-based dual color fluorescence in situ hybridization assay with TMPRSS2 and ERG probes on LN-cap cells derived from the murine air pouch. They found that breaks in the TMPRSS2 and ERG locus of LN-CAP cells derived from B6 mice, but not in SCID mice. And they think this is the consequence of host inflammation. Second, they found that frequencies of inflammation-induced in vivo breaks were higher at the TMPRSS2 locus compared to the ERG locus. And they think it's the DNA breaks in ERG are a rate-limiting step. Then they test the role of macrophages in LPS-mediated TMPRSS2 and ERG gene fusion formation in the big 6 mouse air pouch model. It found that macrophage depletion results in impaired recruitment of other immune cells to air pouch. And by using encapsule as control and clodrosome as macrophage depleted setting, it clearly indicates a role for macrophages or neutrophils in TMPRSS2 and ERG gene fusion formation. So here's some conclusions. First, because SCID mice contain macrophages, so macrophages are necessary but not sufficient for in vivo gene fusion formation. Second, bromo domain and extra-terminal domain BET protein inhibitors like JQ1 has been shown to decrease pro-inflammatory cytokine production in macrophages and ablate inflammatory response in mouse models. Third, treatment with JQ1 blocks TMPRSS2 ERG gene fusion formation in the air pouch model for in vivo inflammation. And fourth, JQ1 also blocks AR function, androgen receptor function. It is conceivable that the observed effects are due to a combined suppression of inflammation and androgen signaling. So according to all the results of the test, the author suggests that androgen receptor induce the proximity of genes such as TMPRSS2 and ERG, while inflammatory stimuli trigger DNA breaks by oxidative stress. And their studies implicate microhomology-mediated non-homologous end-joining as the repair process contributing to inflammation-induced de novo genomic rearrangements. And this study suggests that anti-inflammatory may have a role in the chemoprevention of prostate cancer. And second, there is a potential role for inflammation-induced oxidative stress in the genesis of gene fusion across different cancer types. So this is the end of my presentation.
Thank you very much. So, are there any questions from Kyoto side? Oh, yeah, one question from Kyoto side.
Sorry, I didn't completely understand the slides about the macrophage.
This one?
Yeah, the next one, please. Yeah. What's the function of those experiment groups like encapsule, clodrosome, and different treatments? Sorry, I didn't get it.
Okay. The encapsule is the control group that's just without macrophage depletion, and clodrosome is an anti-osteoblast drug, and in this study is the macrophage depletion settings. So if macrophage is not depleted, then there will be TMPRSS2 and ERG gene fusion. But if the macrophage is depleted, then there will not this result. And treatment with JQ1, I'm sorry, maybe I didn't explain this. This is the protein, and it will block TMPRSS2 and ERG gene fusion formation because it will suppressing the inflammation. So if we treat with JQ1, then it will block the TMPRSS2 and ERG gene fusion. So do I answer your question?
Okay, thank you.
Thank you. Is there? The fields of these genes, and in previous studies, they find that in prostate cancer, it is an indicator of the prostate cancer. And I've searched the function of this gene. I didn't find a clear function of it, but I think maybe it's because the fusion means the abnormal of the gene, and it's like mutation, so it turns out to be the cancer. Is my understanding. Yes. So the normal function of TMPRSS2 is encoding the transmembrane protease, and it's an androgen regulated gene. So it's sensitive to androgen, and the ERG gene is the transcription factor. It will encode the transcription factor. Because here is the normal, the TMPRSS2, and here's the ERG. So you can see that the TMPRSS2, only the exon 1 is fused with the ERG 4, 5, 6, and 11 exons. So I think the function may just depleted, and there may not be a normal protein with function. Any questions?
Yeah, related to this panel on this slide. So, can I ask what percentage of the cells underwent gene fusions?
Excuse me, please repeat the question, please.
The fusion rate of the cells treated with TNF-alpha. What percentage of the cells have those gene fusions?
It's about 50%. 50, yeah, 50. Half,
okay.
just the half.
So related to this paper, so the different cell types with the inflammation go through some specific gene fusions?
You mean the?
Because different cell types have different transcription patterns, so that means different part of the chromosomes are opened. Not only prostate cells, but different tissues have different type of gene fusions after inflammation?
I'm so sorry, I'm not clearly understand the question.
Or the prostate cancer cell line, you see 50% of the after TNF treatment, right? This is very, very high, okay? And that is how they can do the experiment. Very low, it's very hard to do the experiment. But in different type of cells, for example, in the stomach cells, were they creating a similar infusion? Because different gene expression patterns in different. So what do you think about it?
I think I might have to sorry, just I think I'm not quite clear about that, so I think the specific cells may have their own type of gene fusion, perhaps.
I think what Chisaka-sensei is getting at is the cell line itself, LNCAP, that's already a prostate cancer cell line. It's already transformed. So it's going to have some genomic instability already built into it. I think that's part of why the frequency is so high. But to get back to TK, if you had ordinary like HEK293 cells, would you expect the same frequency of fusions for them?
And it comes to the energy. So all these genes are actually one thing I would like to and also advise to the students is that if you are speaking in public,
Yes. No, but thank you very much for your good suggestions. Questions?
Okay, so thank you for listening. Okay, thank you.
Yeah. Thank you. Thank you very much. Bye.